Myself, Professor Sujata Mahapatra from the Department of Botany, RDWU University. In this video lecture, I shall be discussing about transcriptional regulation in the E. coli, and as an example, I shall discuss the operation of the lag operon. Transcriptional regulation is a part of BSc and different MSc syllabi of this university. Transcription in prokaryotes involves a double-stranded DNA, the enzyme RNA polymerase, promoter, operator, regulatory cis and trans-acting elements, regulatory proteins, and the information flows from DNA to RNA in case of E. coli. Cis and trans-acting elements. Cis-acting regulatory elements control gene expression and they are present in the same DNA as the gene. Trans-acting elements control gene expression from a different DNA from a distance. Operon concept was proposed in the year 1961 by two French microbiologists, Francois Jacob and Jacques Monod, and they received Nobel Prize along with Andrew Loaf in the year 1965. This concept, operon concept, explains gene regulation in E. coli. This is the picture of Jacob and Mrs. Monod. In E. coli, lag operon is involved in the transport and metabolism of the disaccharide lactose. Operon is defined as a stretch of DNA that includes the structural genes, promoter sequence, and the operator sequence. And these are a group of contiguous genes because they are placed near to each other. Structural genes are the genes which code for a functional product, either a polypeptide or a protein. Structural genes may be of two types: housekeeping genes, which undergo constitutive expression and they are expressed inside the cell at a constant rate, and the regulatory genes are expressed under induction as per the metabolic requirement of the cell. The lag operon is under the control of these sequences or these elements: promoter, operator, structural genes, repressor gene, and cap binding site. Promoter, it is cis-acting because it is in the same DNA as the gene. Promoter is the sequence to which the enzyme RNA polymerase binds. RNA polymerase is coded for by another DNA, so it is a trans-acting substance. Operator. Operator is the sequence to which the repressor binds, and there are two types of operator, lag operators, primary operator and two pseudo operators. The primary operator is a palindromic sequence of 26 base pair length. Lag repressor binds to this sequence when lactose is not metabolized. Structural genes ZYA, they code for three types of proteins or polypeptides. Z codes for the enzyme beta-galactosidase, Y for the symporter or the membrane transporter galactoside polymerase, A for transacetylase. The repressor gene I is under the control of a independent promoter which is different from the lag promoter, and the repressor gene undergoes constitutive expression to give rise to the repressor protein which binds to the operator, and when repressor protein binds to the operator in the absence of lactose, it protects the operator against the nucleus dilatation, and this occurs when there is no inducer. When there is no inducer, the operator site is unprotected, and when repressor binds, it stops gene expression as well as it protects the operator sequence against nuclease digestion. Cap binding site is the site for the binding of the positive regulatory element cyclic AMP cap complex. Lactose, it is a disaccharide. Glucose is a monosaccharide. So E. coli cell prefer glucose over lactose. But when glucose is not available, lactose is available, then only the lac operon is on and lactose breaks down to produce glucose and galactose, and then glucose is metabolized by the cell. For metabolism of lactose, first the lactose has to enter into the E. coli cell, and this is assisted by the symporter, the membrane transporter, beta-galactoside permease. It is a H plus beta-galactoside symporter that carries a proton and the lactose at the same time. Beta-galactoside permease is coded for by the gene Y. After entering into the cell, lactose breaks down to glucose and galactose or allolactose, and this is catalyzed by the enzyme beta-galactosidase that is coded for by the gene Z. Third enzyme that is involved in the lactose metabolism is beta-transacetylase. It has no direct role in the metabolism of lactose, but it has a role in transferring the acetyl group from acetyl coenzyme A to beta-galactoside, and it degrades the unutilized beta-galactosides. This is the cell membrane of E. coli cell. This is the cytosol. The lactose enters into the E. coli through the symporter, galactoside permease. These are the membrane transporter integral proteins. After entering into the E. coli cell, lactose is converted to allolactose and lactose is broken down to glucose and galactose in the presence of the enzyme beta-galactosidase. Lac operon is an example of inducible operon because it is activated in the presence of an inducer, that is lactose. In presence of lactose only, the lac operon is switched on. Lac operon can undergo negative regulation as well as positive regulation. The binding of repressor stops or switches off the lac operon, that is called as negative regulation. Positive regulation, the binding of CAP, catabolite activator protein, increases the expression of the lac operon multifold. That is called as positive regulation. So, lac is an inducible operon that can undergo both negative as well as positive regulation. These are the genes present in the lac operon. Not I. I is outside the lac operon, so it is indicated by this gap. There is a promoter sequence, operator sequence, and three structural genes Z, Y, and A. I gene is not a part of the lac operon, but it controls the negative regulation of the lac operon. It codes for the repressor, and there is a separate promoter under which the I gene is present. Cap binding site is the site for the binding of cyclic AMP and cap. P is the promoter sequence for three structural genes Z, Y, and A. And The enzyme RNA polymerase binds to this promoter sequence. O is the operator sequence to which the repressor binds, and the three structural genes Z, Y, and A transcribe a polycistronic mRNA. The polycistronic mRNA undergoes splicing to produce three segments. Each segment codes for one polypeptide. One codes for beta-galactoside permease, one is beta-galactosidase, and third codes for transacetylase. Gene RE codes for the repressor protein, and it is under the control of a separate promoter, a different promoter, independent of the lag promoter. It undergoes constitutive expression and it codes for the regulator protein, the repressor protein that controls the regulation of expression of the genes Z, Y, and A. CAP site is located upstream to the promoter sequence. It is the site or sequence to which the cyclic AMP CAP complex binds. CAP is catabolite activator protein and it is a positive regulatory elements of the lag operon. This is the lag promoter sequence of E. coli. It is located upstream to the operator sequence and the operator overlaps the promoter. Therefore, when the repressor binds to the operator, RNA polymerase cannot bind to the promoter as the lag promoter is blocked by the binding of the repressor to the operator. The cap binding site where the cyclic AMP cap binds is located upstream to the lag promoter sequence. The lag promoter sequence is a cis-acting element of the lag operon. These are the sequence of lag operator site and cap binding site. Cap binding site is upstream to the lag operator site. The lag operator, this is the primary lag operator, and it is a palindromic sequence, and the length is 26 base pairs. Lag repressor binds to this lag operator when lactose is not metabolized. Operator is a cis-acting element because it is in the same DNA as the gene. Cap binding site is the binding site for the cyclic AMP cap complex. Lag repressor is a tetrameric protein. It has 4 subunits. And this diagram shows the binding between the lag repressor and the operator sequence. There are two pseudo operators O1 and O2 and two DNA binding domains of the lag repressor bind to two pseudo operators, and that produces a loop-like structure of the DNA, and this is the position of the Y gene and the Z gene after the looping is over. This diagram shows the interaction between the DNA and the lag repressor. These are the two alpha helices of the lag repressor protein, and these two alpha helices are joined by a short stretch of amino acids. And this is the major group of DNA, and this is alpha helix turn. Helix turn means this is the turn, helix turn helix motif, and this is the interaction between the repressor protein and the major group of DNA. This diagram It shows the binding of the tetrameric LAC repressor to the major groove of DNA. LAC repressor binds to the major groove of DNA and the inducer allolactose binds to the LAC repressor. As it is an allosteric protein, it has separate binding sites through which it binds both to the DNA at the operator sequence and to the LAC inducer. And when the inducer binds, there is conformational change of the LAC repressor and it dissociates from the major group of the DNA. Negative regulation of the LAC. Negative regulation means binding of the repressor switches off the operon. And this happens when there is no lactose or lactose is not metabolized because of the availability of glucose. Negative regulation occurs in a stepwise manner. So when lactose is not available, the I gene, as we discussed earlier, always codes for a protein. It undergoes constitutive expression. And the repressor protein, under the situation when there is no lactose or glucose is available, binds to the operator by helix-turn helix interaction. In case of wild type E. coli, the number of repressor protein at any time is as many as 10 copies. Repressor protein is a homotetramer and it is an allosteric protein. It has binding sites to bind to the operator as well as to the inducer, allolactose. That is why it is called as allosteric. Repressor binds to the palindromic sequence of 22 base pairs. Binding of DNA to the subunits of the repressor is by helix-turn helix motif, and there is a strong and tight association between the operator and repressor. So, unless bound by the inducer, the repressor binds to the operator. And when repressor binds to the operator, the RNA polymerase cannot bind to the promoter because in the lac operon of E. coli, the operator sequence overlaps the promoter sequence. Now, what happens when the inducer binds to the repressor and under what circumstance the inducer binds? Inducer binds when glucose is not available and lactose is available. Under this circumstance, allolactose, which is the inducer, binds to the repressor. In E. coli, allolactose is always present in the cell at a lower concentration. So when needed, allolactose binds to the repressor. Binding of allolactose to the repressor brings about a conformational change, and in the changed conformation, repressor cannot bind to the operator, so it dissociates from the operator. So this diagram shows how the repressor dissociates by the binding of the inducer, and after dissociation, there is separation of the repressor and the allolactose. When the repressor dissociates from the operator, the promoter becomes free and the enzyme RNA polymerase binds to the promoter. After binding, it initiates the transcription of the genes Z, Y, and A, and there is Synthesis of a polycistronic mRNA. The polycistronic mRNA undergoes splicing to produce 3 fragments. Each fragment gives rise to one polypeptide after translation. So Z gives rise to beta-galactosidase, the enzyme that breaks down lactose to glucose and galactose. Y gene codes for the protein, the galactoside permease that permits lactose into the E. coli cell, and transacetylase is also produced. Positive regulation of the lag operon is achieved by the catabolite activator protein cyclic AMP complex. Cyclic AMP concentration in the E. coli cell is low when the glucose level in the medium is high. When the glucose concentration in the medium goes down, cyclic AMP concentration inside the cell increases. When the concentration of cyclic AMP is high, it binds to the cap and it forms a complex between cyclic AMP cap. Once the complex is produced, it binds to the cap site, and binding of the complex increases the expression of the lag operon almost 50 folds. CAP, or the catabolite activator protein, is a homodimer. It is made up of 2 similar subunits. It has affinity for cyclic AMP when CAMP level is high inside the cell. And once CAP binds to cyclic AMP, the complex goes to the cap site and binds here. This is the cap site. It occurs at minus 61.5 with reference to the start of transcription site. Cap binding increases the expression of the genes Z, Y, and A many times. It has been calculated that the gene expression increases 50-fold by the binding of cap cyclic AMP. The lag promoter is a weak promoter, and when the RNA polymerase binds to it, only RNA polymerase binds, the rate of gene expression is very low. But once cyclic AMP cap binds, the rate of gene expression increases multifold. Cap cyclic AMP can increase the expression of genes under the control of galactose operon, arabinose operon also. Such type of regulation by a common regulator is seen in E. coli, and this type of complex is called as a regulon. Lag operon is an inducible operon, and it is under the control of the negative regulatory element, that is the repressor, and positively regulatory element, that is cap. The negative and the positive regulatory elements act in a concerted manner. Cap is positive regulatory element, but it will increase the gene expression only if the repressor is not associated with the operator. So this is called as concerted operation of the lag repressor and the cap. And this type of gene regulation is found in the wild type E. coli. If there is a mutation of the I gene, different types of results can be seen. If the I gene is mutated and do not produce the repressor, then there is constitutive expression of the lag genes. And if the I gene is active, produces a repressor, but the repressor is not capable of binding to the operator, then also there is constitutive expression of the lag gene in the absence of an inducer. Mutation of the operator locus is another condition that prevents the binding of the normal repressor to the operator and that also results in constitutive expression of the lag genes. Thank you.